Preparing the fetal pig for dissection. In this video, we will identify the surface anatomy structures of male and female fetal pigs, review directional terminology, and demonstrate proper technique for skin and superficial fascia removal. Let's review the surface anatomy of the fetal pig. The main regions of the body are the head, the trunk, which comprises most of the body, including the appendages, and the neck, which is located between the head and trunk. The head includes the mouth, external nares or nostrils, which are located at the end of the snout or rostrum, auricle or external ear, eyelids, and nictitating membrane. The nictitating membrane keeps the eyeball clean by separating the upper and lower eyelids and is visible when the eyelids are pulled apart. The trunk includes the anterior thorax that is enclosed by ribs, posterior abdomen, anus, tail, and appendages, which include forelimbs and hind limbs. Note that the toes or digits end in hooves and that the first toe is absent. Now, reposition the fetal pig to view the ventral or front surface and examine the posterior or back region of the fetal pig. Regardless of whether you have a male or female fetal pig, you can identify mammary papilla on the ventral surface of the trunk. In females, 5 to 7 pairs of these structures are associated with mammary glands. They have no known function in males. Identify the umbilical cord on the abdomen, which attaches the fetus to the placenta in a sow. Now, let's review the surface anatomy differences between male and female fetal pigs. Male specimens have a preputial orifice, which is the opening to the penis, just posterior or toward the tail to the umbilical cord. This is the opening of the urethra, which releases both urine and semen in adult male boars. Also, the scrotal sacs that will eventually hold the testes appear as swollen regions near the anus. Female specimens have a protrusion just in front of the anus called the genital papilla. This is a common opening for the urethra and the vagina of sows. To review, the surface anatomy structures are head, neck, trunk, mouth, external nares, auricle, eyelid, nictitating membrane, thorax, abdomen, anus, tail, appendages, umbilical cord, mammary papilla, and preputial orifice and scrotal sac in the male, and genital papilla in the female. Now let's review directional terms, which describe the relative locations of body parts. It's important to note that anatomical position for a four-legged animal is with all 4 limbs on the ground. Cranial or anterior means toward the head, and caudal or posterior means toward the tail. Dorsal or superior means on the surface of the back or toward the back, and ventral or inferior means on the surface of the belly or toward the belly. Proximal means closer to the point of origin, which is usually the trunk, and distal means farther from the point of origin, usually the trunk. Rostral refers to a point closer to the tip of the nose. The last set of directional terms is demonstrated with the fetal pig lying on the dorsal or ventral side. Medial means closer to the midline, which is an imaginary line that divides the animal into left and right halves, and lateral means farther from the midline of the body. To review, directional terms are cranial or anterior, caudal or posterior, dorsal or superior, ventral or inferior, proximal, distal, rostral, medial, and lateral. Now let's review the proper technique for skin and superficial fascia removal. First, secure the appendages by tying string to one forelimb and one hind limb, passing the strings under the dissection tray, and tying the strings to the other forelimb and hind limb. This will make it easier to make the proper incisions. Use the scalpel to make a short, shallow incision at the base of the neck. Now, use the scissors to cut through the skin to the top of the umbilical cord. Always point the scissors at an angle away from the underlying tissues as you cut. Cut around both sides of the umbilical cord and continue both parallel incisions toward the anus. Make an incision from each parallel incision down the medial surface of each hind limb to the hoof and cut the skin completely around the ankles. Now return to the base of the neck and cut anteriorly toward the mouth and make lateral incisions beneath the chin. From the midline incision between the forelimbs, make lateral incisions to the hoof and cut the skin completely around the ankles. To remove the skin, use the blunt probe to tease the skin away from underlying muscles. Begin at the posterior end of the fetal pig, where the underlying tissues are less delicate, and work toward the neck. You will tear the cottony connective tissue fibers as you remove the skin. Work from the ventral surface around the lateral surface toward the dorsal surface. Pull the skin over each limb. Remove the skin from the last limb and gently separate the skin from the fetal pig. You may notice muscle fibers attached to the skin. These are cutaneous muscles that allow the pig to twitch the skin to remove irritants. Once the skin has been removed, use forceps to gently remove as much adipose and other connective tissue as possible to expose the skeletal muscles. Once both sides of the fetal pig have been cleaned of these connective tissues, you are ready to dissect the skeletal muscles. Let's review preparing the fetal pig for dissection. The surface anatomy structures are head, neck, trunk, mouth, external nares, auricle, eyelid, nictitating membrane, thorax, abdomen, anus, tail, appendages, umbilical cord, mammary papilla, and preputial orifice and scrotal sac in the male, and genital papilla in the female. Directional terms are cranial or anterior, caudal or posterior, dorsal or superior, ventral or inferior, proximal, distal, rostral, medial, and lateral. In this video, we will identify the digestive structures of the head, oral cavity, and thorax in the fetal pig. Identify the digestive organs and structures of the abdominal cavity in the fetal pig, and compare homologous structures in the human. First, let's review the digestive structures of the head of the fetal pig. The skin and underlying connective tissue on the side of the head have been removed to identify the three salivary glands: the parotid gland, submandibular gland, and sublingual gland. Next, we will identify the structures of the oral cavity. The mandible has been cut and separated from the maxilla to open the oral cavity. Note the incompletely developed teeth of the fetal pig. The hard palate and soft palate form the dorsal surface of the oral cavity and lead to the oropharynx. The muscular tongue houses taste buds and helps manipulate food in the mouth. When swallowing, the epiglottis closes the respiratory passages. Food moves through the oropharynx to the esophagus, which is posterior to the larynx and trachea. These are respiratory structures and are identified here for clarity. The esophagus travels through the thoracic cavity and penetrates the diaphragm. To review, digestive structures of the head, oral cavity, and thorax are the parotid, submandibular, and sublingual salivary glands, hard palate, soft palate, oropharynx, tongue, epiglottis, and esophagus. The parotid, submandibular, and sublingual salivary glands are homologous in the human, as are the digestive structures of the oral cavity: hard palate, soft palate, oropharynx, tongue, and epiglottis, and the esophagus in the thorax. Now let's review the digestive organs and structures of the abdominal cavity. To visualize all of the organs, it is necessary to move the intestines out of the abdominal cavity and to retract the liver. Note the spleen on the left lateral side of the organs, just caudal to the stomach. This is a lymphoid organ only mentioned here for clarity. Let's follow the path of food from the esophagus as it penetrates the diaphragm and moves to the stomach at the cardia region. The next region of the stomach is the fundus, followed by the body, then the pylorus, which ends in the pyloric sphincter. When food leaves the stomach, it enters the first part of the small intestine, the duodenum. Most of the length of the small intestine is the jejunum, and the last region of the small intestine is the ileum, which ends in the ileocecal valve. Notice that the small intestine is attached to the dorsal body wall by the membranous mesentery. The small intestine empties into the large intestine at the pouch-shaped cecum. The remainder of the large intestine in the fetal pig is the spiral colon, which ends at the anus. The accessory abdominal organs of the digestive system are the liver, gallbladder, and pancreas, which are located in the upper half of the abdominal cavity. The large reddish-brown liver, located just posterior to the diaphragm, has 5 lobes in the fetal pig. One liver function is the production of bile, which emulsifies dietary fat. The liver is attached superiorly to the abdominal wall by the falciform ligament. The gallbladder is visualized when the liver is retracted cranially. The gallbladder is green because it stores bile produced in the liver. The common hepatic duct transports bile from the liver and merges with the cystic duct from the gallbladder to form the common bile duct that empties into the duodenum. The last digestive organ in the abdomen is the pancreas, which is located within the curved portion of the duodenum under the stomach. The pancreas produces digestive enzymes, known as pancreatic juice, and connects to the duodenum through the pancreatic duct. To review, digestive organs and structures of the abdominal cavity are the stomach, small intestine, large intestine, liver, gallbladder, common hepatic duct, cystic duct, common bile duct, and pancreas. The spleen is labeled for clarification. The stomach, liver, gallbladder, small intestine, and pancreas are homologous in humans. The large intestine of the human includes an appendix and ascending, transverse, descending, and sigmoid regions of the colon. Again, the spleen is noted for reference. The common hepatic duct, cystic duct, and common bile duct are also homologous. Let's review the digestive system. The digestive structures of the head, oral cavity, and thorax are the parotid, submandibular, and sublingual salivary glands, hard palate, soft palate, oropharynx, tongue, epiglottis, and esophagus. The digestive organs and structures of the abdominal cavity are the stomach, small intestine, large intestine, liver, gallbladder, common hepatic duct, cystic duct, common bile duct, and pancreas.